A-Level Biology 9700, Paper 4, Variant 1, October, November 2024, Question 10. And question 10 is, the populations of the moth Biston butyllaria live in Europe and in North America. The most common phenotype on both continents is a pale wing colored with light gray shading the typical form. So the moth phenotype with dark wing color, the melanic form, also occur on both continents. Shows the typical form of the moth, and it shows figure 10.2 shows the melanic form of the moth. Now, scenario about these pictures is the question. The two melanic European moths were crossed together. The wing colors of the offsprings were 15 typical and 41 melanic. It means the 41 melanic shows the uh, quantity of these melanic, that is, they are greater in number. It means they belong to the dominant species. So the typical is only 15. It means they show the recessive organisms. So construct a genetic diagram to explain these results. You may use these uh, symbols capital A and small a to represent the alleles. So this question is a classic example of a genetics problem involving Mendelian inheritance and the famous case of the peppered moth, Biston butyllaria. So peppered moth uh, exist in two main forms, the typical form light color and the melanic form dark color. So this variation in wing color is an example of polymorphism. So they show the polymorphism. So the two melanic European moths, they crossed. So the typical are of light color and the melanic are dark color. So we can say that the phenotypes of these phenotypes are typical and the second one melanic. And the genotype of them as uh, the typical, the capital A word is for dark and small a is for light color. Melanic veil is more common in offspring. So you can say they are greater in number. So it means they are likely to be dominant. So the genotype for melanic can be capital A, or capital A, small a. On the other hand, for typical, it can be only small a, small a. So since two melanic moths produced both melanic and typical offsprings, you may two melanic, both of their melanic European moths were crossed together. They were crossed together. The wing colors of the offsprings were 15 typical and 41 melanic. It means both they were melanic. It means they can be in the phenotypes melanic into melanic. And the phenotypes for them can be capital A, small a, because they are producing both typical and melanic. So the gamete formation, and then with the help of Puneet's scale, we can construct capital A, small a, capital A, small a. So it can be capital A, capital A, capital A, small a, capital A, small a, small a, small a. Capital A, it will represent melanic, 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 and typical. So the genotypes are of offsprings or gametes. They are capital A, capital A, capital A, small a, capital A, small a, small a, small a. And the phenotype, melanic, 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 and typical. So we can see that this is the ratio is 3:1. 3 melanic or 1 typical. So this is the Puneet scale test. You can see with the help of this genetic diagram, we can explain that the capital A is dominant over the small a. So capital A will represent to the melanic because they are greater number. Because when two melanic European moths, in case if both of they were dominant in this way, the all which are producing, they can be capital A and small a. But here they have told us that there were the 15 typical. So 15, we need to have 15. So for this, the phenotype which they used, the genotype was capital A, small a, capital A, small a. Because capital A dominant, if single present, it will all show the represent, it can represent itself. Whereas the small or the which shows the recessive ratio, so it will only be appeared when it is in homozygous recessive condition. So here, this is the genetic diagram. Now we are moving towards the next question. And next question is, in a similar experiment, two melanic North American moths were crossed together. The color of the offsprings were 10 typical and 31 melanic. What can be concluded about the melanic that causes the melanic form in the moth populations in both continents? So in this case, the question actually it can relate with the previous genetic problem we have discussed in the peppered moth by comparing results. So here I would like to say the independent assortment uh, implies that the same genetic mechanism, dominant and recessive allele, might be operating in the both European and North American populations of the peppered moth. So similar phenotypic ratio can be observed, which is 3:1. So here you can see the 31 melanic and 10 typical. So suggesting it is suggesting a common pattern of inheritance. So with the results, we can conclude that alleles that causes the melanic form in the moth population is both continents. So we can say that they are we call them as dominant alleles. So dominant allele, which one is dominant? That is melanic. Because they are greater in number. So again, we can see this is the 3:1 ratio. So 3:1 ratio, it means 3 are melanic. Again, the same cross will be here. Same will be here. So melanic is dominant. over typical. Now, next question is researchers did not know if the allele causing the melanic fawn in European moths occurred. Researchers did not know if the allele causing the melanic form in European moths occurred at the same locus as the allele causing the melanic form in the North America moths. To find out, they carried out the following crosses. The cross one, European moths that were heterozygous. Heterozygous at the European melanic locus only were crossed with North American moths that were heterozygous at the North American melanic locus only. So in both cases, there were heterozygous in and melanic, and the cross two, the melanic and the typical offsprings of cross one were mated together. So explain why cross two is the test cross. First of all, we must know what is a test cross. So test cross is a genetic cross. It's a genetic cross which between an individual with unknown genotype. Genotype and with the homozygous recessive. Homozygous dominant and homozygous recessive. Here the question is homozygous recessive. It means small a and small a, same type of genes. Okay. So explain why cross 2 is a test cross. So test cross. In cross 2, the melanic offsprings from cross 1 are being crossed. So they were crossed. We know they are melanic, but we don't know if they are homozygous dominant or heterozygous dominant for the combined melanic alleles. So if they are homozygous recessive, the typical offsprings from cross 1 are homozygous recessive for the typical phenotype. So this is because the excess express the recessive trait. So by crossing the melanic offsprings with the typical offspring, researchers can determine the offspring. If all the offsprings of cross 2 are melanic, then the melanic parent were homozygous dominant. If the offsprings show a 1:1 ratio of the melanic to typical, then the melanic parents were heterozygous. So cross 2 follows the classic test cross setup, where an individual with an unknown genotype, the melanic offspring, is crossed with a homozygous recessive individual, the typical offsprings to reveal the unknown genotype. So here we will write cross 2 follows. Here an individual with an unknown phenotype, melanic offspring, or melanic offspring is crossed with homozygous recessive. As for example, capital A, small a, we will write over here in this way. Capital A, capital uh, yeah, capital A, small a. Sometimes when we are writing, we some problems can create in this way. Uh, and as a result of this, the offsprings which are produced as F in F1, they are capital A, small a. So when we cross capital A, small a with homozygous recessive, they are homozygous dominant. So when we cross the F spring offsprings or with the homozygous recessive parents in this way. So as a result of this, where will be the, so this is called as the test cross. So here in this case, as we are crossing the melanics, we are crossing the melanics with the homozygous recessive. So this is called as the test cross in this case. Next question. Complete table 10.1 shows the predicted results if the European and North American melanic alleles are on the same locus, capital A, small a. And second scenario is the Europeans and North American melanic alleles are on the two different loci. So capital A, small a, capital B, small b. So here we have to complete this. In case of same locus, we can see for genotypes of melanic moths from cross 1. So already you know capital A, capital A, capital A, small a, if they are present on the same locus, so these can be the capital A, capital A, capital. These are the two possible, you can say, genotypes. And on the other hand, the different, if they are different, so we can say that they can be capital A, capital B, small b, and capital A, and small b, small b. So the different, in this case, there can be the different. And in the proportion of the two crosses cross to giving 100% melanic offsprings. If it is giving the 100% melanic offsprings, so proportion of test crosses cross to giving 100% melanic offsprings. So it can be 1 in 3, and whereas different locus, there will be none. There will be nothing. It will be 0. I would like to explain this. In this scenario, since both parents were heterozygous for the same locus, capital A, small a, the melanic offsprings will also be heterozygous. So, on the other hand, proportion of test crosses, you can see giving 100% melanic offsprings. If you cross capital A, small a with small a, you will get a 1:1 ratio of, in this case, capital A, small a, when we cross it. So there can be the 1:1 ratio. How? Let me draw it. So they can be capital A, small a ratio, small a, 2:1 ratio or 1:1 ratio. So in this way, I would like to say, so let me explain this table as well. First of all, I would like to go with same locus. So genotypes of a melanic moths from cross 1. So in this scenario 1, you can see the same locus, the genotypes of melanic moths from cross 1. And cross 1, it is capital A, small a, small a. So these two possibilities can be here. And on the other hand, you can say the proportion of test crosses cross to giving 100% melanic offspring, and it cannot be 100%. So it is 1:3 when it is on the same locus. So genotypes of melanic moths from cross 1 in this scenario, since both parents were heterozygous for the same locus, so the melanic offsprings will also be heterozygous. So proportion of test crosses giving 100% melanic offsprings if you cross capital A, small a, or there can be the 1:1 ratio as I have written over here. So the different locus, capital A, small a. So these are the genotypes for them, as you can see in this scenario too. And there can be the proportion of test crosses giving 100% melanic offspring. It will be none or 0. So therefore, no test crawl will produce 100% melanic offsprings. So in this way, I would like to say that this is the scenario in the both these cases. Now, next question is, a light trap was used to estimate the total size of population of bee tularia in a woodland. These are the moths. So one night, 24 moths were captured. These were marked with a small pot of harmless paint. On night 2, 29 moths. First night they were 24, second night they were 29. Captured, and 8 of these showed a spot of paint. Use the Lincoln index formula provided to calculate the size of the population. So show your working. So show your working here. The question is, they have asked us to apply the Lincoln index formula to estimate the population size of distant butyl area that peppered moth, uh, which is based on recapture data. Capture, recapture data. So understanding the Lincoln index is also known as Peterson index is a method used to estimate the size of an animal population. It is based on the principle that the proportion of marked individual in the second sample should be representative of the proportion of marked individual in the entire population. So this formula is used for this. So here I would like to say the key to symbols, capital N, estimate of population size, number of individuals, annual number of individuals captured in first sample, and number of individuals captured in both marked and unmarked in the second sample, and number of marked individuals recaptured in the second sample. So here you can see the capital, we have to find out the capital N, question mark. Okay. So N1 is equal to 24. This is 24 N1 and 29 is N2. And N2 is 29. And then M2 is number of marked individuals recaptured in the second sample. So the 8 of these showed a spot of paint. So they were recaptured. So M2 is 8. Marked moths recaptured. Marked moths recaptured on night 2. So by applying all these values in this formula, N1 is 24 multiplied by 29 divided by 8. So calculations, 696 by 8 is equal to 87. So we will write 87 over here. So this is all about question number 10. Thanks for watching. Please like, share and subscribe my channel.